Dear students, the aim of today's experiment is determination of molecular size of DNA sample by agarose gel electrophoresis. Let us start with the principle. Agarose gel electrophoresis is a technique that is used to separate charged molecules like proteins and nucleic acids under the influence of electrical field. During electrophoresis, the charged molecules migrate towards the electrodes that carry opposite charge, and smaller molecules migrate faster than larger ones. The distance of migration depends upon the size, charge or conformation of the molecule, gel concentration or pore size, strength of electrical field, and the effect of buffer on electrical conductivity. The technique is routinely used in biochemistry and molecular biology to determine a molecule's size. Let us now see the requirements of gel electrophoresis. The first requirement is gel. Commonly, agarose, a polysaccharide extracted from seaweed, is used at concentration of 0.5% to 3% to resolve DNA fragments from about 0.2 kilobase pair to 50 kilobase pair in size. Higher concentration of agarose forms stiffer gel through which DNA cannot move easily. Purified agarose are commercially available as powder and are insoluble in water or buffer at room temperature, but can be dissolved in boiling water or buffer. The next requirement is the buffers. Two buffers are commonly used together in agarose gel electrophoresis. The first is the electrophoresis buffer. TBE buffer, which includes tris, borate and disodium salt of EDTA, is usually used as electrophoresis buffer to provide ions to conduct the electricity and to maintain the pH at constant value. The next is the loading buffer. Buffers like bromophenol blue, xylene glycerol, etc. contains a dye to assess the speed of the gel running and also makes samples denser than the running buffer so that the samples sink when loaded into the well. Loading buffers carry slight negative charges in neutral buffers and therefore migrate in the same direction as the DNA during electrophoresis. They are also known as tracking buffer or tracking dye. The third requirement is the fluorescent dye. A fluorescent dye like ethidium bromide or ETBr is used to visualize the separated DNA bands. ETBr is a fluorescent molecule that intercalates between the bases of DNA and glows pink when excited by UV light. It may be used by mixing with the gel before electrophoresis, or the gel may be soaked in a solution containing ETBr after electrophoresis. The fourth requirement is the samples. Isolated DNA samples can be used as the sample. The next is the DNA molecular weight marker. DNA molecular weight marker or DNA ladder or DNA standard is a mixture of DNA fragments of known sizes run along with the samples as size references to determine the size of the sample. Commonly, 100 base pair and the 1000 base pair or 1 kbp ladders are used and are commercially available. The next is the electrophoresis apparatus. The basic setup of an electrophoresis apparatus consists of a tank, tray, combs, support and power supply. A tank is a container containing the buffer and is covered by a cover to prevent the evaporation of buffer. There are two types of electrophoresis tanks, horizontal tank and vertical tank. A horizontal tank uses agarose or acrylamide gel as a support media and is used to determine identity of nucleic acids like DNA and RNA, while a vertical tank uses only polyacrylamide gel as a support system and is used in protein identification and DNA sequencing. A tray is a container used as a mold where the gel solution is poured and allowed to solidify to provide shape to the gel as it polymerizes. After solidification and polymerization of the gel, it is submerged in the buffer tank to run the sample. Combs are used to make wells on the gel to load different samples. A support of an electrophoresis apparatus is a small piece of glass or plastic present at the bottom of the tray as a support used during polymerization of the gel. The next is the power supply. The power supply of an electrophoresis apparatus can be monitored and operated in current, which is in amperes, voltage, which is in volts, or power in the form of watts mod, through a power supply and is connected to the tray through a black and red colored cords leading from the power supply to the tray in which the gel is run. The next is the detection system. An ultraviolet light box or trans-illuminator and camera are used as detection system to visualize the bands after running the electrophoresis. When the gel treated with a fluorescent dye is placed on a UV light box, the nucleic acid fragments produce light bands and the sample picture may be acquired by a camera for analysis and documentation. Next, let us see the reagents and buffers. First is 2 liter of 10x TBE buffer. For this, 218 gram of trees base, 110 gram of boric acid, 9.3 gram of EDTA are required. The ingredients are dissolved in 1.9 liter of distilled water and pH is adjusted to about 8.3 using sodium hydroxide and the volume is made up to 2 liter. Let us now see the agarose gel electrophoresis protocol. First, let us mix 0.5 gram of regular agarose powder in 50 ml of 1x TBE buffer and microwave for about 1 minute to dissolve the agarose. Then leave it on the bench for about 5 minutes to let the solution cool down to about 60 degrees Celsius, which is like just hot enough to be able to hold with bare hands. Now add 1 microliter of 10 mg per liter ethidium bromide solution to the gel mixture and swirl to mix the contents. Next, pour the gel into the gel tray and put the combs of required sample size. The gel is allowed to solidify in room temperature for at least 30 minutes. Remove the tray from the support and immerse it in the tank containing 1x TBE buffer. Now mix 10 microliter of DNA with 10 microliter of loading buffer and load it into the well. Then mix 5 microliter of loading buffer with 2 microliter of leather and load it in another well. Now place the lid on the gel box and connect the electrode wires to the power supply correctly, connecting the positive or red and negative or black wires. Run the gel at 100 volts for 20 minutes or until the dye line is approximately 75 to 80% of the way down the gel. The maximum allowed voltage varies depending on the size of the electrophoresis chamber and should not exceed 5 volts per centimeter between electrodes. After the dye line has run approximately 75 to 80% of the way down the gel, turn off the power and disconnect the wires from the power supply. Next, remove the lid of the electrophoresis chamber and carefully remove the tray and gel. Now place the gel on the tray of a trans-illuminator and photograph the picture. Let us now see the results. The bands corresponding to the isolated DNA and the leather are observed in the picture. The purity and quality of the isolated DNA sample is checked by observing the bands. A thick, bright band of the DNA sample represented a non-degraded, good quality X-ray. To calculate the size of bands in the length of the gel, first a standard curve from the migration of the DNA fragments in the 1 kV leather is constructed by plotting the log 10, which includes the number of base pairs for each fragment on the y-axis against the distance that each fragment migrated on the x-axis. The sizes of DNA molecules in the sample can be estimated by interpolating on the standard curve. Alternatively, calculate the RF value using the formula RF equal to distance traveled by DNA molecule over distance traveled by the dye. A standard graph can be obtained by plotting the RF values against the respective molecular size of a DNA marker in a semi-log graph sheet, and the standard graph can be used to determine the molecular size of any linear DNA band electrophoresis on the same gel. Let us now see the conclusion. The molecular size of DNA samples can be determined by agarose gel electrophoresis. Agarose gel electrophoresis provides a means of analyzing DNA by separating molecules based on size. Thank you.